Hi, my name is Jenny and I'll be talking about how muscle coactivation shapes sensory feedback processing during human movement. Our body is capable of learning and performing many complex motor actions, such as running, writing, or reaching for objects. Achieving such coordinated movements, however, depend on how effectively the nervous system processes sensory feedback to guide and correct our motor actions. For example, to avoid getting tackled during rugby. During these motor actions, our muscle follows a reciprocal pattern where the agonist muscle turns on to initiate the movement, where the antagonist muscle remains silent. The antagonist muscle will then turn on to terminate the movement, while the agonist muscle falls silent. But in some of our motor actions, like if we're trying to make precise movements with our hand, then our muscles will actually coactivate. Muscle coactivation refers to the simultaneous activation of the agonist and antagonist muscles with opposing action of the same joint. A long-standing idea is that muscle coactivation helps to increase the stiffness of skeletal muscles, so they become resistant to stretch, helping to stabilize the body and resist physical disturbances that we may experience in everyday life. However, there are instances that this seems a little counterintuitive because some motor actions require us to quickly initiate or correct our movements rather than to maintain stability. To understand this phenomenon, we must go back to the basic neurophysiological functions of muscle coactivation and study the interactions between our body and the environment. This raises the question of how muscle coactivation influences sensory feedback processing and behavioral performance. To answer this question, we conducted an experimental study where we examined the relationship between controlled levels of muscle coactivation, sensory feedback processing, and behavioral performance with healthy controls. We predicted that in healthy controls, higher levels of muscle coactivation would amplify the nervous system's responsiveness to sensory feedback and in turn result in faster corrective responses and improved performance. We had 5 healthy participants perform an upper limb postural control task in a canine exoskeleton robot. Participants were seated in the robot with their dominant arms supported in truss that allowed them to move in the horizontal plane. The robot was also paired with a VR system that allowed the participants to interact with the target via a cursor that was aligned to the fingertip of their index finger. The first task was a normalization task to account for the individual differences in how muscle activity levels relate to the level of force production, controlling muscle coactivation across all my participants. This was achieved through reference loading. Participants were instructed to stabilize their cursor in the target and resist 1 to 5 Nm external loads, while the surface electrodes collected activity from the biceps and the tricep. This task allowed us to collect participants' reference EMG, which was utilized in the experimental task. The experimental task utilized a custom EMG biofeedback program where the participants performed the task under controlled levels of coactivation. During this task, participants were instructed to stabilize their cursor in the target. However, this time they would encounter mechanical perturbations that would rapidly flex or extend their elbow. Participants had to return the feedback cursor into the target within 400 milliseconds after the perturbation for a successful trial. This task design allowed us to examine how mechanical perturbations applied to the elbow affect muscle and kinematic responses. When looking at the results, we first analyzed the elbow displacement. We observed that at higher background loads, participants increased activity in both the elbow flexor and extensor muscles. This increased coactivation stiffens the joint and results in less displacement following perturbation. We also examined the movement time and error rates. We anticipated that as participants increased coactivation, they would be able to correct for perturbations more quickly and more precisely. This would be reflected in shorter movement time to return the cursor to the target in the allotted time, and fewer errors in task performance. Our findings supported this. This suggests that while coactivations can increase muscle stiffness, it can also be leveraged strategically to optimize performance when the task demands speed and precision. Interestingly enough, though, we see this trend change in the 4 and 5 Newton-meter condition. Lastly, looking at the activity of the elbow flexor and extensor muscles following mechanical perturbations, we gain insight into the neural implementations of the task. Side by side, there's an EMG plot showing the flexor and extensor across the short latency response and the long latency response time windows. In the long latency response time window, we've observed increased excitation of the flexor and greater inhibition of the extensor as the background load increased. This indicates that the nervous system becomes more responsive to proprioceptive feedback as coactivation level increases. The shared long latency response in both agonist and antagonist muscles may reflect a deliberate neural strategy to maintain precision during challenging tasks. Traditionally, muscle coactivation has been understood primarily in terms of its mechanical benefits. However, we lack a detailed understanding of how muscle coactivation interacts with neural control of sensory feedback. In our experiment, we observed patterns that challenged these traditional ideas. Participants increased muscle coactivation not just to stabilize the joint, but also in conditions where they needed greater mobility to respond quickly. This suggests that coactivation plays a more dynamic role in motor control and generally supports our hypothesis. We found that great activity of the elbow extensor and flexor muscles was associated with corrections and reduction in peak hand displacement. Taken together, greater muscle coactivation correlates with faster and more vigorous responses to proprioceptive feedback. Here are some references. And finally, I want to thank my supervisor and lab members who helped me with my research.